polypeptide is a singular linear chain of amino acids. The amino acids are linked covalently by polypeptide bonds. These polypeptide chains go on to make proteins essential for growth, repair, structure, function and regulation of the body's tissues and organs. With this, it is important to understand polypeptide synthesis, which simply means the process of making a polypeptide. It is during this where the two main steps occur: transcription and translation. Polypeptide synthesis starts in the nucleus of the cell, the location of the DNA and site of transcription, the process's first step. This DNA is made of phosphate bonds and a sugar called deoxyribose, which both form the backbones, and then 4 nitrogen bases with each pair joined with a hydrogen bond. To begin polypeptide synthesis, the enzyme of helicase unwinds and unzips a portion of the 2 strands of DNA. One of these strands will be used as a template for DNA replication. This small section of DNA has a start and stop signal, used in order to recognise the code needed for a particular protein needed at the time. Complementary RNA nucleotides pair with the chosen DNA strand's bases. Cytosine and guanine nucleotides pair together, and with adenine and thymine nucleotides also pairing. However, anytime RNA places a complement to A on the DNA, it will place uracil instead of thymine. The RNA polymerase enzyme then binds these RNA nucleotides together, making a new single-stranded RNA. This new strand is called messenger RNA or mRNA. This provides the specific codes and information of the exact order of amino acids that need to be bonded together to form a polypeptide. Once formed, the mRNA then travels from the cell's nucleus through the nuclear membrane and into the cytoplasm, where it will attach to a ribosome made of rRNA or ribosomal RNA. Thus begins the next step of translation. In the cytoplasm, there are many tRNA molecules available, also known as transfer RNA, that carry an amino acid, the monomer of a polypeptide. The ribosome within the cytoplasm reads the mRNA code, 3 bases called codons at a time, and adds the complementary anticodon, found on one end of the tRNA. These codons provide the information that determines the related amino acid. The other end of the tRNA carries that related amino acid, integral for the building of a polypeptide. With the first tRNA and amino acid attached, the next tRNA carrying another amino acid will move in and attach its anticodon to the mRNA's complementary codon. The 2 amino acids carried by the tRNA will link together by forming a peptide bond. Then, the first tRNA and its anticodon will detach, leaving behind the amino acid, where it can pick up another amino acid. The mRNA will shift through the ribosome and the next tRNA molecule can come in. As more tRNA anticodons match with the mRNA codons, the amino acid chain becomes longer. This will continue until a stop codon is reached, a codon that does not code for an amino acid. Once the stop codon is read, the amino acid chain detaches and the polypeptide is formed. There are many strengths and weaknesses that can be observed in this model of polypeptide synthesis. An advantage is that this model simplifies the complex process of polypeptide synthesis, putting it into terms which can be interpreted easily. However, in saying this, depending on the level of understanding you wish to achieve, the model may be too oversimplified and lack the specificity of each step within polypeptide synthesis, meaning it is missing detail, causing it to be inaccurate. For example, there is far more detail and complexity to making the mRNA. However, for simplicity, this is left out. Yet, a model is meant to be the simplification of a complex process, and so, this model does show the basics within each step and process of polypeptide synthesis. Another advantage of this model is that each feature is clearly labelled and of a distinguishable colour and shape, enabling better understanding of the process of polypeptide synthesis and the elements involved. Viewers can clearly determine elements due to the label, colour and shape. For example, the tRNA molecules are of a forest green colour and shaped in a cross, unlike any other piece. A disadvantage, however, specific to the model, is that there is no bonds between each nitrogen base pair. After making the DNA, there was not enough room between each pair to add something that will represent a hydrogen bond. Also, it was too difficult to add a bond between the anticodon and codon. This can limit the understanding of how the enzyme of helicase works and how the anticodon attaches to the mRNA's codon.